So I did my research on, I kind of based it on like nutrition and kind of building muscle, breaking down muscle, but specifically in Duchenne muscular dystrophy. It's one of the most common types of dystrophy involving the breakdown of muscle because of a mutation in your gene. And I thought that that was really interesting to study because it's something that people don't talk about, but it's actually really, really common. And so kind of looking at like potential mitigations that can maybe kind of stop like this from happening, possibly some treatment. Right now, there's no cure for it. So it was just really interesting to kind of look at and figure out like what are the differences between the different bodily systems and muscular breakdown and muscular protein synthesis. So those kind of things. So let's jump in. So kind of touched on that. So the first thing that I wanted to do is just have a brief overview of kind of what the different dystrophies are. So I kind of laid out a bunch of them. Duchenne and Becker are very, very similar, but obviously each different dystrophy affects different body parts and causes different muscular wasting. And something interesting that I found was just the effect of like cardiomyopathy and that presence in Duchenne muscular dystrophy. That was a very common thing. I think like 25% of people with Duchenne muscular dystrophy actually run into kind of that issue. And so obviously, like I said, there's no cure, but there's a lot of potential treatments coming out just with gene manipulation and a lot of oral medications. That's kind of the most common thing you'll see with the I can't even pronounce that. So that one. Yeah, also something that I did want to look at though was just the effect of resistance exercise and how that can build muscles. And possibly serve as a potential treatment and just intaking more protein. All right, so for the next kind of section I just wanted to touch on was specifically what Duchenne muscular dystrophy is. So the first thing is like just in terms of prevalence, it's very common, 1 in 5,000 to 6,000 male births typically have it, and people who are male, they typically are affected by it based on the genes because of the X chromosomes. So it's more present in them, but there's obviously just a couple of exceptions where females can get it as well. Like I said, it's very common. Some of its characteristics are just like the cardiac complications. It affects the respiratory system a lot, and patients really barely live past 40, although with increasing treatments, the age at which people can live to and live with a lot of movement capacity has been increasing, so that is a good sign. And then also just like 13 and older, usually wheelchair bound, so non-ambulant, and there's a lot of frequency with a reduced skeletal muscle mass and also a lessened amount of fat-free muscle mass and fat-free mass. So that is something interesting that you can see, and a lot of accumulation of scarred muscle tissue. And something that I found that was super interesting was a lot of times people falsely give credit to Duchenne, but in actuality, it was Edward Miron who had kind of discovered the disorder and characterized it for the first time. And he also found out the symptoms about like cardiomyopathy, so that was really interesting. Yeah, and scientists along with him found that the sacrolemma cell membrane has the primary defect in it. So, all right. And moving on, I kind of wanted to talk just a little bit about the dystrophin glicoprotein complex. So it is just kind of a membrane that houses a lot of different proteins, but the main one is obviously dystrophin because that's the one that we want to focus on, and that's where the defect is in the disease. So dystrophin, it just links the cytoskeletal and membrane elements. It's super interesting. You can kind of see the little wavy line, the blue one, that's kind of where it's connected. If you can't see my mouse moving around. And then also DMD individuals, they just have a lot of impaired signaling muscle regeneration. Some some studies have found that their muscle protein synthesis is lowered, while others have found that their muscle protein breakdown, and we'll get into more of this, is raised. So it's kind of still kind of a questioning area of kind of what's going on, which stops them from building muscle. So yeah. Let's see here. So now I just wanted to talk a little bit about skeletal muscles. So it's obviously super, super important, especially in homeostasis, regulating heat. And it's super important for your metabolism. People always talk about having more muscle stimulates your metabolism, allows you to burn through that fuel faster. So it also regulates posture. It converts chemical energy to mechanical energy. Evidently, it allows you to move. So, and it kind of just gives you a lot of like freedom in movement, making your own decisions and kind of interacting with your environment. Yeah, and it can store fuel. Interesting enough, people always think about fat being the thing that stores all the fuel, but really your muscles can store fuel too. And if you if you're in a mode of starvation, it can actually cause your muscles to break down. So it's kind of interesting. Your body draws from your muscles. Yeah, so let us move on. Something that I wanted to talk about also was just like how skeletal muscle looks. So it's multinucleated post-mitotic, and it can't undergo mitosis, which is kind of like the division of, it's a type of cellular division. So you can kind of see in this picture, the little black things are the nuclei. So showing all those nuclei all over there. It just has connective tissues kind of connecting the muscle to the muscle fibers. Each nucleus is responsible for a specific type of protein in its area or its nuclear domain. So super interesting that each nucleus is delineated for a specific reason. So. All right, so moving on, I kind of wanted to just look a little bit at skeletal muscle mass compared to non-DMD individuals and DMD individuals. So as you can see, total body mass was about 28% for non-DMD individuals. Their body proteins was about 75%, and the protein turnover rate, which is the building muscles, was about 50%. On the other hand, DMD individuals, they had 3% and 5% of muscle mass based on their body weight. Like I said, fat-free mass was a lot lower, and just generally they saw kind of trying to rise from supine, being on your back, there was a longer time for if you had less muscle mass. So it makes sense. And some contractile proteins, the ones listed in them, those were specifically very affected by DMD. So their fractional synthesis rates, or FSRs, were a lot lower, a lot less protein turnover for those. All right, so just generally a key idea is the idea of how your body creates muscle, and it builds muscle when you're in an anabolic or positive NPVAL. Don't ask me what that stands for, I kind of forgot, but basically you have this thing called muscle protein synthesis. It's building muscle, but on the other hand you have muscle protein breakdown. That is the breakdown of muscle. So you can build muscle when you have a greater MPS than MPB, but you lose muscle when you have a greater MPB and MPS. And don't get me wrong, muscle protein breakdown is super, super important for the restructure of muscle, but it is something that is typically people who have DMD are in a catabolic state, meaning that they are breaking down muscle. And also in a fasting state, you're typically breaking down your muscle. So that's interesting to know. So kind of moving on with the building of muscle via the uptake of amino acids. So they are influenced by a variety of feeding strategies. Some studies found that it was really interesting. Americans consume protein in a skewed manner. They consume it mostly at dinner, which isn't a surprise, but they did find that kind of consuming protein regularly in kind of bouts of 20 grams if you're a younger, healthy individual causes you to have a 25% increase in MPS for the 24 hours after you consume it. So kind of those like the times in which you consume protein is a little bit important, but in general, just like the amount of protein you consume is kind of what stimulates the building muscle. They did find that the older you get, the more protein you need in healthy individuals, but especially for DMD individuals, they require even greater intakes of protein to kind of build that muscle. So kind of showing that protein intake might be a possible solution, not the only thing that you can do, but something to help maintain muscle. All right, this is pretty long, so sorry about that. For healthy boys, they kind of looked at how much their muscle protein they were accumulating. It was 0.34 kilograms a year. On the other hand, it was 0.56 kilograms a year for DMD individuals. Like I said, kind of touching on the contractile proteins, DMD individuals had 73% and 89% lower synthesis rates for tendon and myosin light chain. But on the other hand, the sarcoplasmic proteins were not really affected in DMD, and they were typically pretty similar compared to the healthy controls. So that was interesting to know. All right, so some of the ways that scientists study kind of both skeletal muscle mass and muscle protein synthesis is there's a variety of ways, but in my personal opinion, I would say that the D3 creatine dilution and the heavy water labeling are probably the most advanced and beneficial methods for measuring skeletal muscle mass and MPS respectively. And that's because they're non-invasive. For D3 creatine dilution, it just involves ingesting a labeled creatine sample and then collecting a fasted urine sample to figure out how much skeletal muscle mass a person has. On the other hand, MRIs are not the greatest considering the potential of radiation and also just the inability to measure total body muscle mass. You have to measure it in specific sections, and so it's just not the most effective thing. So, but it was interesting to kind of learn about the different ways that scientists have used, especially in the studies that I was reading during my literature review. So. Just a little bit of overview of muscle protein breakdown since we kind of touched on muscle protein synthesis. There's three main systems. The first one is the UPP. That is kind of involved in the protein turnover for all cell types. Autophagy is just used for proteins in exercise performance and adaptation besides the myofibrillar proteins, and the calpains. Those are kind of reserved for what's left over, which is mostly the myofibrillar proteins. So those are all involved in kind of the breakdown of muscles. But I would like to say, kind of just to reiterate, muscle protein breakdown is not bad. It's simply a way for your body to remodel your muscles. And like I said, if you're in a fasted state, it serves as a source of fuel. So it is very important, an important process. Something really cool that I saw, and actually it was in one of my mentors' previous research papers, was the idea of ingestion or like creating insulin. So when you eat, obviously you create insulin, and that is super important for inhibiting muscle protein breakdown. So while you have the amino acids, the proteins, those ingestion can stimulate and create a higher amount of MPS. You have kind of the insulin, which suppresses the MPV, so you have a greater anabolic like state. So it creates kind of that net positive NPV-AL. So that's kind of cool, and it serves as a way to build muscle. So something I found was obviously DMD individuals. Their issue is that they are unable to really exceed their MPV with the MPS, so they are always in a catabolic state, or almost always. So, but it was interesting to kind of see whether studies thought that was because their MPV was raised, or was it because their MPS was lowered? So there was kind of a discrepancy there between articles. All right. So just in conclusion, DMD, it can cause muscular atrophy. It kind of limits the quality of life that people have, and it is honestly one of the most common forms of dystrophy, and something that you might even see in your lifetime. And scientists who just study it have been using non-invasive tracer techniques, like I talked about, the D3 creatine and the heavy water labeling, or the 2H2O is what it's called. But obviously we are looking towards the future, looking at prospective treatments, and some of those treatments I actually skipped over. So we're going to kind of look at those just a little bit more. So just the glucosteroids, those are kind of the main things, but unfortunately they cause a lot of side effects, but they are available in the US, and then the prednisone, or however you pronounce that, it carries a lot less side effects, but a lot of countries have not approved it for treatment. There's non-invasive ventilation, the ACEI, and then you have some therapeutic approaches. The myostatin inhibitors can inhibit MPB, so that's super interesting. And just obviously in taking more protein can help build muscles. And then the resistance-based exercise. If you see on the right, there was a study that kind of measured the effect of resistance-based exercise on how much people would build muscle in DMD. So as you can see, just like, for example, the sit to stand, you had a really great increase from the pre, which is like before the resistance exercise, and from the post, which is 2 weeks after the 12-week resistance exercise program. So that was super interesting, just to see kind of how resistance exercise can help build muscle, not just for healthy individuals, but for DMD patients as well. So yeah, and just really quickly, my mentor was Sophie Joanise, and she was really, really helpful. She actually she's from Manchester University, and really nice that she was able to accommodate my schedule, and really helped me, especially with editing my paper. She had a lot of knowledge on how to format things, references, stuff like that, which I had no clue how to do, and just guiding me through the literature review. I've never done one, and I will probably do a bunch in college, so it was really cool to see, and just expanding on my interests in science, nutrition, and exercise physiology.